information about nematode problem, especially in watermelon production. So if you, some of you might remember that we have done a survey in the watermelon production system, in specifically in southern Georgia. As you can see, those counties in this map, we basically collected with the help of several extension agents, which I really appreciate their help. We actually conducted a survey. We collected around 66 soil sample from different fields, and we basically try to identify what nematode species or genus are important and for watermelon growth. And we found that among different nematode species, root knot nematode, which all of you might be familiar with, is the #1 pest of watermelon production. 50% of those fields that were surveyed were infested with root knot nematode. In this map, you can see those dots in different color. Those are basically the nematode count per 100 cc of soil. For example, the dot in green is the number was between 0 to 10, and the red one is the number of the nematode greater than 100 per 100 cc of soil. So that means that only one location had a high number greater than 100 nematode per 100 cc of soil, and the rest of them are basically between 0 to 100. So as you can see again, those counties that are really are known as the watermelon growing counties, that we have more spread of nematode, root knot nematode in those counties compared to the county more south of Georgia. So when we compare the root knot nematode number or pressure in watermelon field with the other vegetable crops that are grown on plastic, basically the pressure on watermelon is significantly lower compared to the other vegetable crops. In the right, you can see a field of the watermelon. So the damage, that's an infested field with the root knot nematode. But as you can see some yellowing and some necrosis. But other than that, it's hard to basically see the nematode damage. And that's normal because the nematode damage might not be visible unless you ended up with the high number of the nematode in the soil. So the bottom picture is the root knot nematode galling on the roots. As you can see, when you pull up the roots, you can see those galling on the roots due to root knot nematode infection. The galling might be smaller or medium size, and this is actually the huge gall that I just put here so that you can basically be familiar with the shape of the galls. So this is the root knot nematode. For you, you might be interested. What is the nematode? This is the shape of the nematode. So this is on the left is a second stage juvenile, which is an infective stage of the nematode that basically enter the roots and cause the damage. At the end of the growing season, the nematode basically changed to the female, which is a lemon shape. You can see the green picture. And then at the bottom, you can see the nematode basically produce egg mass on the surface of the soil. That egg mass basically contains between 100 to a couple of hundred eggs, as you can see in this picture. The right-hand side is the adult female, and the left is basically the egg mass, which contains too many eggs. So one of the basically, that means that the gall of the root, galling on the roots is important for us to basically evaluate the damage caused by root knot nematode on not only watermelon, but also other crops, vegetable crops, even row crops. So when for any experiment where we are dealing with the root knot nematode, root galling is the #1, is one of the important things that we look at. What we do basically, there are different scaling or rating procedure for evaluating the damage caused by root knot nematode on the roots. This is one of them, where we actually give the galling a scale of 0 to 5. 0 means there was no gall produced on the roots, as you can see in the picture, and the 5 means that more than 75% of the roots were gall, as you can see in the picture on the left, on the bottom left right hand corner. You can see the differences. Basically, when we are doing any experiment, looking at the nematicide, looking at the resistant root stock, we basically try to measure what the gall index based on these scaling. And we will use this, and you can, I'm just put this slide here because I'm going to talk about some of the data on the nematicide or root stock that you can basically find it out what I'm talking about. So when we talk about the root knot nematode, so there is a direct relationship between the number of the nematode in the soil and the gall index formation. The lower the nematode number, the lower would be the gall index, and vice versa. Here in an experiment, we basically infected the watermelon variety sugar baby with a different population density of the root knot nematode. As you can see, we inoculate with 500, 1,000, 2,000, 4,000, 8,000, and 16,000 of the juvenile per pot or per only one seedling. And we grow the plant for 35 days. And again, we pull up the roots or wash the roots and just look at the gall index. Again, using the here, oh, there's a mistake here. It should be 0 to 5. But as you can see, with the increase in the number of the nematode, you can see the gall index will be increased. So basically, when you have a high number of the nematode in the soil, the gall index will be higher. That means that you will have more damage on the roots. Here in this picture, again, you can see the control on the left-hand side and the 1600 nematode roots that were infested with 1600 nematode on the right-hand side, and you can see with the increase in the number of the nematode, the growth of the roots will be reduced. And as you can see on the right hand side, the roots is completely destroyed by the nematode, and you can see the gall on the roots. Again, you can see in this graph, again, with the increase in the number of the nematode per pot or in the soil, the root growth will be reduced. Here's the for plant high or plant growth. As you can see, again, with the increase in the number of the nematode, the growth of the plant, either both height and the weight of the plant will be reduced. You can see it's really visible here, as you can see. So moving forward, so when we did the survey, as you can see in the first slide, we basically had 50% of the field infested with root knot nematode. What we wanted to basically find out later on was to see what species of root knot nematode are prevalent in the watermelon fields in southern Georgia. We use both molecular techniques as well as morphological techniques to find out the species. And as you can see, Melodigon incognita, which is known as the southern root knot nematode, was the most prevalent, is the most prevalent species of root knot nematode, followed by Melodigon arenaria or peanut root knot nematode, and Melodigon javanica was the less prevalent species of root knot nematode. We also found that almost 22% of the field survey had the mixture of Melodigon incognita and Melodigon arenaria. So that means that some of the field are just infected with only one species, but we also have some field that are infested with 2 species of root knot nematode. So there are a couple of techniques for management of root knot nematode, especially in watermelon, soil fumigation using resistant varieties, which there is no really resistant in watermelon, use of non-fumigant nematicide, cover cropping with suppressive cover crops, use of resistant rootstock for grafting, as well as crop rotation. I'm going to just talk about the trial that we did on grafting and non-fumigant nematicide. So when it comes to rootstock or grafting, well, basically there is no commercial varieties of watermelon with resistant to root knot nematode in the market. However, research that has been done in the last couple of years have shown that there are some accession or some line of the citrullus species that had shown moderate resistance to two species of root knot nematode, Melodon incognita and Melodon arenaria. But honestly, these lines or accession, whatever you want to call them, they are not of interest for the breeders. It might because of it is not only because of the root knot nematode resistance. It's related to other aspects of the plant, including the resistance, whether that plant or that varieties or accession has a resistant to other soil-borne pathogen, including Fusarium wilt of watermelon. As you can see here, there are a couple of rootstock that have a great resistance or tolerance to Fusarium wilt of watermelon. Especially two races of race 1 and race 2. For example, in trispecific hybrid squash and bottle gourd, these two rootstock are resistant to Fusarium wilt, but they are susceptible to root knot nematode. In recent year, basically, I think in 2018 or 2019, there was a paper out from South Carolina, and they reported another rootstock called a hybrid citron that has shown moderate resistance to Melodon incognita, but good resistance to Fusarium wilt. We tried to do a greenhouse test to see how these actually rootstock, especially the intraspecific hybrid squash, can show resistance or susceptibility to infection with both root knot nematode and Fusarium wilt races 1 and 2. In a greenhouse test, we actually had 9 treatments. So we had the Melodon incognita, only the pods were infected with the Melodon incognita. We had the Melodon arenaria. We inoculate 4,000 nematode or J2 of these two species per pot. We also had the Fusarium race 1, Fusarium race 2 as a single application. We also had the combination of either of species of root knot nematode with races of the Fusarium wilt. For example, Melodon incognita race 1, Melodon incognita race 2, Melodon arenaria race 1 and Melodon arenaria race 2, and also we had an untreated core or non-infected plants. So we grown the plants for 35 days in the greenhouse, and then we look at some of the growth factor of the watermelon. As you can see, as I told you already in the last slide, I told you that the intraspecific hybrid squash is susceptible to root knot nematode, and we confirm that here actually. Sorry, that should be the next step. But in terms of the growth, the root weight, you can see that both the plant infected with the root knot nematode had a less root weight compared to the uninoculated or non-infected plant. In terms of the vineless and shoot weight, you can see that the Melodon arenaria reduce these two factors significantly compared to the healthy plants. However, there was no difference between the incognito and arenaria for both shoot weight and vine length. When it comes to fusarium wilt, so as I said, this rootstock is resistant to to fusarium wilt, and as you can see, with regard to the vine length and shoot weight, there is no different. There is no difference between the plant treated with infected with the fusarium wilt compared to the healthy plant. However, we noticed that the plant infected with the both races of fusarium wilt had a less weight compared to the lower weight compared to the healthy plant, which is understandable because we are basically infecting the plant, and the plant basically have some these pathogens have some effect on the growth of the roots. But interestingly, as you can see, the bottom part of this table, we notice there is no interaction between the fusarium wilt 1 and 2 with both species of root knot nematode. That means that when we have a infection with these two pathogens, root knot nematode and fusarium wilt, you won't basically see any damage in terms of the root weight, vine length, and shoot weight due to the disease complex. Okay, but look at the nematode reproduction. Here is again the gall index, 0 to 5. We notice that again when we had the plant only infected with the root knot nematode, Melodon incognita, and Melodon arenaria, we had the gall index of 3. Okay, that means that the nematode, a single application or single infection of the plants with the nematode result in the gall index, high gall index. But when we had the combination of the root knot nematode and fusarium wilt, race 1 and race 2, we still saw that the nematode were able to basically grow in the plants, and the gall index, although is significantly lower compared to the single application of the root knot nematode, but that shows that the nematode were still able to develop and cause gall on the roots. Let's look at the egg mass or egg counts. Again, the same story. So root knot nematode, as when it was infected, when the plants were infected with the only root knot nematode, both incognita and arenaria species, they were able to increase or reproduce on the plant. Okay, this is the number multiplied by 1,000. Okay, you need to multiply these number that are shown in this graph by 1,000. Okay, that means that a lot of nematode were able to eggs were produced by either of these two species. But when it comes to the combination, when we had the disease complex, incognita and fusarium wilt race 1, race 2, arenaria race 1, race 2, and you still can see that the nematode were able to increase its population in the roots. And this is not this shows that, okay, so use of the resistant varieties of resistant rootstock is okay for the field that are only infected with the fusarium wilt, race 1 and race 2. You can go ahead and use the resistant varieties for grafted on the watermelon, grafted on either bottle gourd or intraspecific hybrid squash or hybrid citron. You're fine. You have no issue when it comes to fusarium wilt. But you have to be careful if your field has an infestation is infected with both pathogen, root knot nematode and fusarium wheel, you might need to consider use of nematicide. Basically, you can't really rely on the resistant rootstock. You need to use nematicide to control root knot nematode, okay? So we are going to basically conduct a trial, hopefully this year, to see how nematicide basically help to control root knot nematode when we use grafted watermelon. Okay, so hopefully next year I will give you some more information about this. So let's move forward and looking at some data for the nematicide trial that we did. Here, this is the data for 2019 that we had a nematicide trial with the Velon Prime, only non-fumigant nematicide. We had the treatment Velon Prime at 6.4 ounce per acre, which was applied at transplant. We also had a split application of Velon Prime at 3.25 ounce. The split application was were applied at transplant. And the second application was done 4 weeks post-planting. We also had the Nimitz at the 90 ounce, which was applied 1 week before transplanting. We had the Vidate at transplant, and we had the split application of Vidate at 32 ounce per acre, which was applied at transplant and 4 weeks after planting and untreated check. Let's look at the data. Here's the gall index. Again, the gall index of 0 to 5. As you can see in this column, Nimitz and split application of Vidate, they had a better control of gall index or compared to the untreated check, as well as the single application of Vidate. However, there is no difference between these two treatments and Velon, okay, either single application or split application. When it comes to the nematode count per 100 cc of soil, Nimitz did a better job compared to the untreated check, but there is no difference between Nimitz, significant difference between Nimitz and other treatment, okay? When it comes to the yield, you can see this is the yield, the pounds per plot. In this trial in 2019, basically we rely on natural pollination by bees, but we had not good yield data. But as you can see, for the yield that we had in that year, again, Vidate, single application of Vidate had a numerically higher yield compared to the other treatment, but there was no significant difference between all the treatment. Look at the Nimitz, okay? Look at the Nimitz. Nimitz had a lower yield compared to the numerically compared to the other treatment. Let's move forward with the 2020 trial. In this past year, we actually had again the same treatment. We had the single application of Velon, split application of Velon. We had the Nimitz. We had the single application of Vidate, and also we had the split application of Vidate. Now look at the data. In this trial, we found that split application of Vidate followed by Nimitz, they had a significantly lower gall index compared to the untreated check. However, there was no difference between these two treatments with the other treatment, okay? In terms of the nematode count per 100 cc of soil, Nimitz followed by split application of Vidate did a better job, especially when it was compared to the untreated check. But there is no difference between these nematicide trial. Significant difference, okay? Let's look at the yield. In this trial, we actually include some pollinator in the plot. So we basically use the pollinator varieties SP7 in the trial, and then we had a better yield. You can see in this column, but in terms of the efficacy of nematicide on the yield, you can see the split application of vellum voided. Oh, sorry. Okay, there is a difference between this trial and the last trial. As you can see, let me. In 2019, we had 2 applications of voided, whereas in 2020 we had 3 applications of voided. 1 at transplant, and the other 2 were applied with a 2 weeks interval. Okay. This is the only difference between this trial and the 2019 trial. But in terms of the nematode count, again, nimids followed by voided, they did a better job in terms of the nematode control, but there is no difference between the nematicide. Look at the yield. The yield, 3 application of voided per season had resulted in a significantly higher yield compared to the nimids. I show you in the last slide that nimids had a lower yield compared to the other treatment, compared to the other nematicide trial. That means that it seems that nimids have some adverse effect on the yield of the watermelon. You notice that in terms of the nematode control, nimid was found to be a good nematicide. It's a good nematicide, but when it comes to the yield, we found that in the last 2 years, we found that the nimid had some adverse effect on the yield. So the other conclusion is that the single application of vellum prime at plant, as well as split application of voided during the growing season, especially when you apply 3 application of voided during the growing season, you basically can not only control root knot nematode, but also can expect more watermelon yield. That's all what I have for today. I also would like to add that we publish an extension bulletin talking about management of root knot nematode in watermelon. You can easily search these either at the UGA extension website, or you can even Google it, and you can find it, and you can basically find out more information about different management technique for root knot nematode in watermelon. With that being said, I'd like to thank you everyone, and let me know if you have any questions.